Great, let's get started. Hello, everyone. Welcome to Acrobiosis' webinar on how TRFRET can empower drug screening and mechanism studies. My name is Lisa Cho, and I'm currently the Product Marketing Specialist at Acrobiosystems, and I'll be your host for this webinar. And before we start the presentation, please feel free to take a photo of this QR code for a quick TRFRET questionnaire to fill out at your own time, and that way you'll also get an Acro gift of your choice. And as for today's agenda, we'll have a 20-minute pre-recorded talk by Yusan, who is our VP of Product Development at Acrobiosystems. And with over 12 years of experience in life sciences, Yusan has been driving innovation by building scalable platforms that accelerate antibody discovery and development. And during the presentation, feel free to type your question in the Q&A box. And then at the end of the presentation, we'll have a live Q&A session with An Oyan, who is our product development manager overlooking Acro product lines in the North American region. And he collaborates closely with the PD, marketing, and sales team at Acro to ensure our product and services address the unmet needs of our customers in various areas in CGT, antibody, drug development, and CMC manufacturing. And without further ado, let's start with Yusan's presentation to learn more about TRFRET.
Good morning, everyone. It is a pleasure to have this opportunity to share with you all the application of TRFRET in drug development. In the next 30 minutes or so, I will introduce the invention of TRFRET, how this technology being used in research, and a few case studies of TRFRET in drug development. So first, a little bit about ourselves. We're called Acrobiosystems. We were founded in 2010 and went public in 2021. We currently have 900 employees all over the globe to support the pharmaceuticals and institutes for drug discovery and production work. We offer a wide variety of tools with a particular focus on recombinant proteins. In today's talk, I'd like to introduce our new series of product, TRFRET kits. The history of TRFRET dated all the way back to 1948, when German physical chemist Theodor Foster discovered the resonant energy transfer, a mechanism describing energy transfer between two light-sensitive molecules. 30 years later, the second piece of this technology was discovered, and that is time-resolved fluorescence. TFR takes the advantage of long lifetime of fluorophore to avoid interference caused by fluorophore with a short fluorescence lifetime. The fluorophore of choice are usually chelates of lanthanides, most commonly europium and terbium. With the breakthrough of cryptand, TRFRET technology was quickly developed as an assay method, and it was adopted in different types of drug screenings, such as GPCRs, kinase, antibody drugs. In the recent years, the application of TRFRET technology has extended into microRNA detection, biosensing, cell function measurement, and many more. Now let's have a closer look at this technology. First, Fluorescence Resonance Energy Transfer, FRET. FRET involves two fluorophore, a donor and an acceptor. The requirement is that the emission spectrum of the donor overlaps with the absorption spectrum of the acceptor. Excitation of the donor by an energy source, such as a laser, produces an energy transfer to the acceptor if the two are within a given proximity to each other. The efficiency of this energy transfer is inversely disproportional to the distance between donor and acceptor, making FRET extremely sensitive to small changes in distance. In practice, when distance is over 10 nanometer, only donor signal is detected. When the distance becomes less than 10 nanometer, FRET occurs, acceptor signal is also detected. As I mentioned in the previous slide, the choice of donor is usually chelates of lanthanides, most commonly europium and terbium. That is because lanthanides have a very long fluorescent lifetime at a millisecond level, whereas others' lifetime are at 100 nanosecond level. From the example on the left, we can see signal of a regular fluorophore quenched quickly, whereas signals of lanthanide last for a very long time. This time difference lays the foundation of time-resolved fluorescence. How does it work? So when the signal is collected in a sample using FRET method, a time delay is set to 50 to 200 microseconds, which means the signal will be only collected after 200 microseconds upon donor excitation. The organic signal of acceptor is quickly quenched, and due to the long lifetime of lanthanides, the signal detected at this time are only donor signal and energy transfer from donor to acceptor, which significantly reduce the background noise. The resulting assay provides an increase in flexibility, reliability, sensitivity, in addition to higher throughput capability and fewer false positive and false negative results. The sensitivity of TR-FRET reaction is at picomolar level, which requires only 2 to 10 microliters of samples, super friendly for rare samples such as precious serum samples. The method is very fast. It takes about an hour from sample prep to signal collection. It is compatible with different types of testing candidates, such as nucleic acid, small molecule, antibody, serum, cell, and etc. A liquid system makes it very easy to operate and doesn't require any wash steps. It is also easy to automate to achieve high throughput demand. Since the reaction takes place in a homogeneous liquid system, all epitope of the molecule are exposed, resulting in fewer false, positive, and negative results. Because of these advantages, TR-FRET technology has been used in many types of applications, such as bending assays, cell function, protein quantifications, and many more. And now we will take 3 examples to see how TR-FRET can be used in these aspects. First, bending assay-based inhibitor screening. KEAP1 and NRF2 pathway is a very important antioxidant pathway. Under basal condition, KEAP1 binds to NRF2, targeting NRF2 for degradation. Under stress, the structure of KEAP1 changes and loses the ability to bind to NRF2, which leads to accumulation of NRF2 in cytoplasm and translocation of NRF2 into the nucleus. In turn, induces the transcription of cytoprotective genes. This pathway is often targeted in treating inflammation, CODP, and other diseases. The inhibitor targeting KEAP1 NRF2 can be screened using TR-FRET method, wherein KEAP1 protein is conjugated to a donor by a tagged antibody, and NRF peptide is conjugated to a FITC. Naturally, KEAP1 binds to NRF2, brings the 2 fluorophore together. FRET occurs. However, upon adding the inhibitor, the interaction between the 2 is disrupted. No FRET signal can be collected. In this assay, the interaction of KEAP1 and NRF2 shows a dose-dependent manner. As the concentration of NRF2 rises, the binding affinity increases, reflected by the FRET signal. Inhibitors targeting this interaction can be screened using this mechanism. In this example, 2 compounds are tested using TR-FRET method. Both interrupt the interaction between the 2, showing by the decrease of FRET signal. And compound number 2 showed a significantly lower IC50, suggesting more powerful inhibitory effect over compound number 1. In addition to bending assays, TR-FRET can be used in all to assess cell functions. GLP-1 receptor is a member of the GPCR family. It has been a very hot target for studying diabetes and obesity. The endocytosis of GLP-1 receptor is crucial for insulin release and beta cell homeostasis. Using FRET, we can monitor the endocytosis of GLP-1 receptor in live imaging. A snap tag links the donor to GLP-1 receptor on the cell surface. At the same time, non-permeable acceptor is added into the reaction. When GLP-1 receptor is on the cell surface, donor and acceptor are close enough, FRET happens. However, when the GLP receptor is internalized upon stimulation, distance enlarged, FRET disappear. 3 ligands are selected to test their effect on GLP-1 receptor endocytosis. As we can see, they all exhibit ability to induce dose-dependent receptor endocytosis reflected by FRET signal. As concentration rises, internalization increases. Among the 2 ligands, GLP-1 and exidin-4 showed better effect on receptor endocytosis compared to other ligands. TR-FRET can also be used in quantification assays. As we all know, cytokines are key modulators for immunity and inflammation, and they are important biomarkers for a variety of diseases. Quantitative measurement of cytokines allows the assessment of immune status, monitoring the immune responses, and the investigation of pathological processes. We can build a TR-FRET reaction using double sandwich method. Takes IL-6 as an example. 2 antibodies that bind to IL-6 are conjugated to donor and acceptor respectively. When IL-6 is added into the testing sample, the binding complex will bring the donor and acceptor close together. FRET happens. Using this mechanism, a standard curve can be generated based on IL-6 with a known concentration. The concentration correlates very well with the FRET signal. Using this standard curve, we can quantify the amount of unknown IL-6 in the sample. Here at Acro, we developed a variety of different TR-FRET kits to support your drug discovery work. I'd like to use a few cases to show you how we build our assay and why you should choose Acro's TR-FRET tools. The first case study is the inhibitor screening kit for TL1A. TL1A, DR3, DCR3 is a very important pathway to modulate inflammation and homeostasis. High expression of TL1A has been shown in IBD, RA, and cirrhosis. Recently, multiple IBD drugs targeting TL1A has shown positive results in clinical studies, which makes TL1A a very hot target for IBD and other autoimmune diseases. We've developed TL1A inhibitor screening kits where we conjugate a donor and an acceptor to TL1A and DR3. Upon the presence of an inhibitor, the interaction between TL1A and DR3 is interrupted, hence there is no FRET signal, which is a perfect method for high sensitivity inhibitor screening. Though the concept is simple, the success of this assay depends on the high-quality TL1A protein being used. The natural conformation of the TL1A is a heterotrimer. In solution, TL1A exhibits as a mixture of monomer, trimer, and aggregates. How to obtain a trimer form that is close to its natural conformation is a challenge. One common approach is to induce mutations in the sequence to obtain stable monomer. However, that would change the conformation of this protein, led to inaccurate results for drug screening. That's of course not the way we want to go. We then optimize different conditions in production, such as culture medium formulation, fermentation, purification, and eventually we obtained a TL1A trimer with a high purity without altering any natural sequence. This TL1A exhibits natural conformation in solution with high bioactivities, and it's used in our TR-FRET inhibitor screening kits. Using protein with a natural conformation ensures the high fidelity of the testing results and reduces the low potential candidates pathing to downstream assays. The TL1A inhibitor screening kits are also validated by at least 2 commercial available TL1A drugs. Both show the dose-dependent FRET signal, suggesting this kit is perfect option for TL1A inhibitor screening. In fact, we have developed a portfolio of inhibitor screening kits to cover hot therapeutic targets in autoimmune disease and oncology, and more to come before the end of the year. These are all complete kits and ready to use. When you don't really have an option for a complete kit, you can also build a TR-FRET assay using our DIY toolbox. Again, take TL1A inhibitor screening as an example. The toolbox will provide you with a donor-acceptor conjugated anti-tag antibodies or streptavidin, and you need to add a tagged TL1A and DR3 proteins to build a quaternary system. In addition to using high-quality proteins, as we discussed before, the key to success of this assay is to have the compatible anti-tag antibody to bind to the labeled target proteins. His-tag proteins are commonly used in research. However, the length of his tag can be different, and the accessibility of his tag can also be different. Therefore, anti-his antibody we choose must be highly compatible with potential binding partners. As you can see in the graph on the right, the anti-his antibody in our toolbox has been tested against different types of his-tagged proteins. It has been proven to be highly compatible with all sorts of binding partners. Here is an example where we use our toolbox to build a inhibitor screening kit targeting the PD-1 and PD-L1. In this assay, we use a biotin-related PD-1, his-tagged PD-L1, donor-labeled streptavidin, and acceptor-labeled anti-his antibody. We can see in the graph on the right, when we increase the PD-1 antibody added into the system, FRET signal is quenched, suggesting the inhibition of the interaction. Aside from his-tag antibody I mentioned, we also provide donor-acceptor conjugated antibody against other tags, including FC, GST, FLAG, as our DIY toolbox. Like the anti-his antibody, these toolboxes are validated to be highly compatible with different tagged proteins. Another application of TR-FRET is FcRn binding assay, which is an important assay in drug development. The FcRn binding affinity is highly dependent on pH in the system. We tested the FcRn TR-FRET kit in different pH, and as the pH increases, IC50 rises, suggesting the interaction between IgG and FcRn decreases, and they are unable to prevent the FRET from happening. We eventually selected pH 6 for FcRn kits. In the example on the right, we validated these kits using 3 commercial drugs, and TR-FRET signal correlates very well with the SPR results, as well as published half-life of these 3 drugs, suggesting TR-FRET is a reliable method to test FcRn binding, reflecting the lifetime of antibody drugs. In drug development, ADCC is one of the key MOA for many therapeutic antibody drugs, especially anti-cancer antibodies. Researchers design antibodies to intentionally enhance or harness the ADCC effect to achieve better therapeutic outcomes. One approach is Fc engineering, including glycosylation, modification, or set-directed mutagenesis, which can significantly increase the antibody affinity for Fc gamma receptor, thereby enhancing the ADCC effect. This effect can also be measured using TR-FRET. In fact, we tested 4 commercial drugs with different IgG subtypes using our TR-FRET ADCC kit, and they exhibit different level of TR-FRET signal, suggesting different affinities to Fc gamma receptors. This kit is also validated against different culture medias to ensure its compatibility with different types of samples. Here is a list of our FcRn binding kits, as well as the Fc gamma receptor binding kits. All our TR-FRET kits have gone through stringent QC testing to ensure batch-to-batch consistency, stability, and reproducibility. All kits can be shipped with methodology validation reports. And hopefully in the past 30 minutes, I have explained to you the variety of different applications of TR-FRET technologies and have convinced you that AcroBio system is your best choice for TR-FRET kits. As our kits are built with high-quality proteins, highly compatible anti-tag antibodies validated by multiple commercial drugs, and have gone through most stringent QC testing to provide with you the most reliable testing results. Please visit our TR-FRET webpage for more detail. And thank you for listening.
Thank you, Yu, for the presentation. I hope everyone's able to understand more about TR-FRETs from Yu's talk. And again,
good morning.
Sorry about that. And again, if you want an acro gift, please feel free to take a chance to fill out this quick questionnaire regarding TR-FRETs by scanning this QR code. And now I will open the floor for any questions. Let me check the Q&A box real quick. Feel free to type in your questions in the Q&A box, and I can also start with a quick question for An. And what advantage does TerraFred actually offers compared to the common ELISAs?
So the TerraFred, can you hear me, Lisa? Hello?
Hello?
Hello, can you hear me?
Yeah.
Okay. Yeah, the advantage of the TerraFred and versus the ELISA, and so definitely the first is so from the process or steps, and the TerraFred is not required to wash the plate, and it is the homogeneous assay format, and so these will enables the high throughput and save the time, and also the Sun you mentioned in the webinar that the sample volume is quite small and it's only required 10 microliter, and that is the significantly lower than the like the conventional the ELISA, and additionally, and the TerraFred, and it is the it is very compatible and with different sample types, and with like the serum, plasma, and different culture medium and different buffer reagent, and so this is the like the significant advantages and of the TerraFred and versus the conventional ELISA.
Gotcha. Thank you so much. And again, feel free to type in your question or raise your hand, and I can also unmute you to speak up. And I do have another question, like for the TerraFred detection, for do you guys have a specific requirement for the plate reader, and can like the conventional microplate readers be used directly?
Yeah, so that's the question, and so typically the scientists when they learned or something new technology, and the other things that the lab need to consider is the equipment, and TerraFred is the associated equipment is not like very fancy, and so typically the microplate reader and which is equipped with the TerraFred module, like or at least it has the wavelength, fluorescence wavelength. For our products, like the emission wavelength, that's the 620, and the excitation wavelength, that's 655, 655 nanometer, and these wavelengths will be enabled the scientist to read out the TerraFred, the results. So no special equipment required, just either it's equipped with the module or either the machine itself is able to detect the fluorescent signals. So it's a quite, the equipment friendly and the lab friendly, and unlike like the flow cytometry or like the PCRs.
Gotcha. That makes a lot more sense. I don't see any questions yet coming in. I do have another question. How
Okay, sure.
how can the accuracy and the reliability of TerraFreds be detected or like be ensured?
Yeah, okay. So so this is a kind of the very like the big question. So just want to try to be more like general and summarized. And so the factors that affect the assay performance, and it will be multiple and not just a single one. And so tend to seeing the TerraFred, and in the previous questions I mentioned the advantages and the advantages of the TerraFred versus ELISA. And if from the mechanisms of the TerraFred, and it is the time resolved and the acquisitions, and so this will significantly reduce the short-lived, the background of noisy, and versus like typically, and people use the flow cytometry and detect the fluorescent signal. So that is the advantage of TerraFred. And also besides the mechanisms and the reagent is the quite critical and crucial to the assays. And TerraFred requires the two molecules and to be close enough and to stimulate the donor will stimulate the receptor and to detect the fluorescent signal. So that means either the proteins or the antibodies, and they will need to maintain the native conformation and also make sure that their itself, the biological activity, will not be changed after labeled with the metal ions and also the fluorophore dyes. So the so that means find a reliable and high reputation of the reagent supplier, and that is quite important. And also, since the TRFRET is from the principal sites, and it's a little bit complicated versus ELISA. So that's the the consistency or reproducibility of the reagent is very important, especially supports the drug design or drug development that to last for more than a decade. So this is the second part. And the third part is whether the completed the kit or the assay, the panel, it has the appropriate, the positive and negative controls. So that will, help or support the scientist to make judgment whether the assay failed is because of the performance of the reagent or performance of the sample itself, or whether it is because of the human error during the implementation of the procedures. So that's the third part. And so all these three, I will list it as the top three factors that will impact the TRFRET, the performance. And otherwise, like the buffer, the sample, and also the pH conditions, and for others are not like the important versus the previous three factors.
Gotcha. Thank you for the extensive answer. And we do have, I see two questions in the Q&A box right now. The first one is asking about if the sample protein is only biotinylated at the N or C terminus, does it still work with the TRFRET, or do you need a more extensive coverage?
So the biotinylated protein, when it labeled with the C or N, and it could help. It could be used in the TRFRET, and it is so we will have the especially the assay design and the validation to make sure that the donor can stimulate the receptor and to stimulate the excitation wavelengths. So it do not, the 100 required that the biotin should be labeled, the specific, sites or random labeling, and to increase the biotin-related ratio of the protein.
Thank you, An, and I hope that answers your question. And Simon asked in the presentation, we mentioned that the TRFRET sensitivity range is in the picometers. Just in general, like what is our sensitivity in terms like compared to others?
So TRFRET is the very comparable or exactly the very similar the sensitivity or dynamic range and versus the ELISA and the multiplex like the cell-based array and also for others like the SPR no, sorry, not for SPR, and for the BRI and for the specific purpose like to detect the binding affinity, and it is definitely cannot be ethos or translate the results from TRFRET to SPR. And for like some cytokine detections and some like the specific like clinical biomarkers detections, and we have to admit the MSD will provide the highest sensitivity. And otherwise, the TRFRET is very comparable and to the ELISA and to other detection methods.
I hope, Simon, that answers your question. We do have another one asking about for smaller proteins, like around 10 kilodalton, and highly positive charged proteins. Is there any specific protocols or any modifications that may be needed for TRFRET?
No, the 10 kilodalton is not that small. It's still to be incorporated into the TRFRET assays and don't need the specific the charge or any modifications on the protein itself.
I hope that answers your question. And if there are no more live questions about TRFRET, I will I will end the live Q&A session, but again, feel free to stay connected with either me or on with our emails here. Feel free to reach out to us for any questions you may have about the webinar or the TRFRET products. We'll be happy to help out and also connect you to the right regional manager if necessary. And yeah, and thank you again, everyone, for staying till the end of this webinar. And I will now conclude our session and have a wonderful day. Thank you, everyone.